Hi everyone, welcome back. I want to continue with chapter 8. That looks better. Okay. So the first slide here talks about humoral and cell-mediated immunity. So there's two different types of immunity that we have. And again, I don't want to get too detailed because it's just not, what the point is, this class is, but I just kind of want to give you an overview. So humoral and cell-mediated immunity are both necessary. They complement each other. Pretty much the humoral immunity is mediated by the B cells, and the cell-mediated immunity is mediated by the T cells. And both of them identify antigens. Like one of them can eliminate tumor cells. The cell mediate can eliminate tumor cells. The humoral does not. The cell mediated has a delayed response. The humoral has a quick response, and you can kind of see right here the B cells on this side, the plasma cells, the memory cells. Cell mediated is the T helper and the killer T cells to activate the T cells. They remember the cells. Just to keep in mind that we need both to have a proper acquired immunity. Okay, so now we're kind of talking about the acquired immunity with the T cells and the B cells. And that is our third line of defense right here. And it's specific. Remember, 1 and 2 were not specific. Specific meaning it's specific for the specific cells for specific pathogens. I had mentioned this earlier. So there is a specific cell for the chickenpox virus that you have, the flu virus, the pneumonia virus. So one specific cell for each one. And it's acquired. We acquire this in our lifetime. So they're specific and they can only recognize like one single antigen. Lymphocytes are a form of white blood cells that take out specific pathogens. Like I said, they can only recognize a single antigen. And then the lymphocytes have a memory. Both need to see the pathogen first before they respond. Okay. Both develop memories to what they have destroyed. So if it comes back in the body, invades the body again, they remember and have the antibody for it. So the two cells, the T cells, which we talked about, is made, it's kind of easy, made in the thymus gland, can recognize what's wrong. Cells that target and destroy, kills virus infected and damaged cell, the killer cells, cytotoxic cells, and the helper T cells. They help the T cells and the B cells, so this helps the B cells. And B, I kind of remember, B cells produce antibodies. B cells produce antibodies. So it's just a little role that they do. This is a killer, this is a helper, and this is an antibody producer. B cells are made in the bone marrow. B cell bone marrow produces antibodies, which are proteins that bind to foreign material. We're going to talk about this CD molecules right here. These are surface molecules that are expressed on immune cells, particularly the most common are the CD8 and the CD4, and they play a role in communicating. So these cells can communicate amongst each other. So these little markers, they're just essential for identifying and isolating lymphocytes and leukocytes into subsets. And you've probably heard CD8, CD4, and those are kind of a categorization of the T cells. Again, we're not going to be immunologists here, but just some terminology that is helpful to know. Okay, so with the 3 levels of defense that we have, as you know, diseases and infections still happen, okay? So hence we have an immune system. So adaptive immunity can be acquired actively or passively, okay? So this is probably just a review, but it's a good review. Active immunity is immunity that is gained by actively engaging with the antigen through invasion or a vaccination. For example, you get a disease and you build a resistance to a disease. A disease invaded your body. This process occurs, your immune system comes to help, and then you build resistance. You have memory cells in there, so you don't get it again, fighting off a cold. So the next one is passive immunity, immunity that's gained by receiving antibodies made outside the body by another person, animal, or recombinant DNA. And an example of passive immunity would be breastfeeding, passing the immunity on from mother to baby. And these are just kind of some examples of active immunity. We have natural and artificial, and passive immunity, natural and artificial. So the active immunity that's gained by engaging with an antigen, remember, through invasion, either with an infection, which would be a natural way to get it, or with a vaccination, which would be an artificial way of getting it. But still, these are 2 ways to get active immunity. Passive immunity occurs, again, an example, a natural way that passive immunity is gained is through maternal antibodies through breastfeeding, and a passive immunity by passively giving somebody antibodies. We can give people monoclonal antibodies that we inject these antibodies in them, and they're artificial antibodies that we can give them for various illnesses that they may have. So T cells and B cells, we've talked a little bit about those. So just know that the T cells are the killer T cells and the helper T cells. So helper and killer T cells. Here's the helper. Cytotoxic is a killer. So the killer T cells wipe out infections. They wipe out cancer cells. The helper T cell helps that killer's T cell and helps the B cell. And the B cell is here to produce antibodies. So we have the virus that invades the cell, and we have these antigens, and the T cells and the B cells attach themselves, and that's how they kill, and our immunity, how to kill this virus or this pathogen that has invaded us. B cells attack invaders outside the cells, and T cells attack infected cells, kills cells. The T cells killer cells, and the B cell kills them by producing antibodies outside the cell. Okay, here's a slide for the CD molecules, and CD is an abbreviation for cluster of differentiation, and they're surface, like I said kind of earlier, surface molecules expressed on the cells of the immune system that play key roles in immune cell-to-cell communication, just kind of like I showed you that previous picture. These molecules are essential markers for the identification and isolation of leukocytes and lymphocyte different subsets. So really, the only thing you really need to kind of think about with this is the 2 most common are the CD4 and the CD8. And the T cell CD4 is right here, and the CD8 is right here. These are the helper T cells, and they help the body fight infection. You don't need to know any more than that. Okay, the last slide, immunoglobulins or antibodies. There's 5 class. Each of the immunoglobulin class functions in a different type of like an immune response to the body defenses. So these are antibodies that we have in our body that fights invaders. And there's 5 of them, the IgG, the IgM, the IgA, the IgD, and the IgE. So if a person is deficient in any of these antibodies, they're going to be more prone to getting an infection. So these antibodies have specificity for a specific antigen. So the IgGs are specific for a specific antigen. The IgG is the most abundant and accounts for most of our protective activity against infections. So we can transfuse some IgG to a patient through IVs subcutaneously. They get these IgG infusions every week to build up their antibodies because they may be immunodeficient for some reason. You don't have to know these subclasses. So then the IgM produced first upon antigen invasion, and it increases transiently. So basically the IgG and the IgM is abundant as well. IgA is found in blood and body secretion. And again, these react to certain types of pathogens that we're just not going to memorize. You should just really know these right here. Don't know much about the IgD. The IgG is the most abundant. And the other thing that's important to know is the IgE is involved in allergy. So with this IgE, it can help when allergens enter in our body. But sometimes we're going to talk about hypersensitivities coming up. Sometimes somebody will have a hypersensitivity response to pets or some type of an allergen, and they get terrible symptoms like itching and asthma and stuff like that, and they can kind of have like a hypersensitivity or an overreaction. Like too much of this would occur, and then it could cause somebody to have like a hypersensitivity, and you have to treat that. But what you need to know is IgEs are involved, antibodies involved in allergy. Thank you.